Hello, everyone. I'm David Eidelman, Vice Principal of Health Affairs and Dean of the Faculty of Medicine at McGill University. Au nom de l'Université McGill et sa faculté de médecine, bienvenue à tous. Welcome to the third annual McGill Clinical Innovation Competition Finals. And of course, to our inaugural virtual edition, like everything else, we've had to go virtual. C'est un grand plaisir d'avoir parmi nous du gouvernement du Québec, M. Pierre Fitzgibbon, ministre de l'Économie et de l'Innovation, qui nous dira quelques mots sous peu. On a aussi M. Paul l'archevêque, dirigeant du Bureau de l'Innovation en santé et services sociaux, qui assiste à l'événement en direct. Merci d'être avec nous aujourd'hui. This competition and the prize given out this evening aim to promote our innovative thinkers within the Faculty of Medicine and Health Sciences and across McGill. Our goal is to inspire individuals and teams to imagine new devices, diagnostics, platforms, programs, and processes to improve healthcare in Quebec, in Canada, and around the world. We aim to enable innovators to turn their ideas into actions and outcomes they can commercialize and implement. There exists a major need for innovation in clinical care, and supporting medical innovation is more important now than ever before. As the COVID-19 pandemic progressed over the last many weeks, we questioned whether this competition should go ahead, but we didn't question for long. In the spirit of innovation, the CLIC team under Jerry Freed, Jake Barrett, and Kevin LaChapelle launched into creating this online version, and as so many have done to step up to the challenges in the faculty, university, government, and industry throughout this crisis. So kudos and thanks to all involved and to everyone joining us tonight, colleagues, students, partners, and innovative thinkers from the private sector. Not surprisingly, the quality of the proposals received this year clearly demonstrates we have the potential to make tremendous impact with the right vision and with the right supports in place. This competition has been extremely competitive so far, and so my congratulations to everyone, including all of our finalists who we will meet shortly. So now I have the pleasure of introducing you to our extraordinary panel of judges from academia and industry, and thank you for your fantastic engagement and for raising this event to such a high standard. So our judges include Nadine Afari, Steve Arliss, Carmela DeLuca, Nicola Franco, Raymond HAKIM, Stuart Kozlik, Michael Mee, Brent Norton, and Dan Roden. We'd like to acknowledge the many excellent judges who were involved in the preliminary judging process as well, whose names you should see here, and thank you all for your expertise. I also want to thank our generous sponsors, Novo, Fasken, the Steinberg Center for Simulation and Interactive Learning, and the McGill-Dobson Center for Entrepreneurship. Thank you for your support of this year's event, details which we'll hear more about later on. And of utmost importance, I want to thank the people behind each of our prizes for their vision, inspiration, and commitment to clinical innovation, especially during these times. MARIKA ZELENKA Roy, in partnership with the Montreal General Hospital Foundation, Don Shepard from the McGill Interdisciplinary Initiative in Infection and Immunity, otherwise known as MI4, and supported by the Doggone Foundation, and Dr. Ray HAKIM, creator of the HAKIM Family Innovation Prize and the inspiration for this event. Maintenant, c'est pour moi un grand plaisir de vous présenter M. Pierre Fitzgibbon, ministre de l'Économie et de l'Innovation du Québec. Et avant ça, avant de se lancer en politique, M. Fitzgibbon était associé directeur chez partenaire Walter Capital. C'est un leader réputé dans le domaine des finances, du développement d'entreprises et du développement des affaires. M. Fitzgibbon a siégé au conseil d'administration de plusieurs compagnies et organisations d'en vigueur, dont la Caisse des dépôts et placements du Québec, WSP et Transcontinental. Je laisse maintenant la parole à M. Fitzgibbon pour le mot d'ouverture officiel de la finale 2020 du Concours d'innovation clinique de McGill. Merci.
Merci beaucoup, David. Alors, mes salutations à Dr. Haldeman. Je sais que le Dr. Quirillon est ici, le scientifique en chef du Québec, que j'ai entendu M. Paul l'archevêque du Bureau d'innovation. Alors, mesdames, messieurs, je suis très heureux d'être avec vous pour ce concours virtuel. Vous en êtes tous très conscients, nous vivons actuellement une situation exceptionnelle et sans précédent sur le plan sanitaire, technologique, économique, social et politique. Bien que le Québec ait été en pause pendant plusieurs semaines, la reprise de l'économie s'amorce de façon très graduelle. On ne peut pas parler de relance et de croissance économique sans parler de recherche et d'innovation, incluant la recherche fondamentale, la recherche appliquée, la valorisation des découvertes et les collaborations internationales. Notre gouvernement va donc redoubler d'efforts pour attirer les meilleurs chercheurs au monde, donner accès aux centres de recherche publics ainsi qu'aux entreprises et les infrastructures de classe mondiale. On va aussi contribuer au développement d'entreprises émergentes, notamment en rendant du capital de risque disponible pour appuyer la croissance et soutenir les incubateurs et les accélérateurs spécialisés. La communauté scientifique québécoise est d'ailleurs très proactive dans la recherche des solutions à la crise de la COVID-19. Nous avons mis en place un processus accéléré pour évaluer les projets, les vaccins, détection de virus, répercussions en santé mentale, et j'en passe. C'est très stimulant de voir la réponse des chercheurs et des organisations scientifiques au Québec. C'est aussi très stimulant de voir aujourd'hui ce concours d'innovation se tenir à McGill. C'est avec l'innovation que les entreprises québécoises pourront d'une part augmenter leur productivité, se forger un avantage concurrentiel et prendre une longueur d'avance dans leurs marchés respectifs. Je rappelle que l'innovation est un pilier incontournable qui va permettre au Québec de réaliser ses objectifs économiques qui sont d'accélérer les investissements des entreprises, d'attirer davantage d'investissements étrangers et d'augmenter nos exportations et d'en diversifier les destinations. On le sait trop bien maintenant, des ajustements doivent être apportés aux soins de santé qui sont donnés à nos personnes les plus vulnérables ainsi qu'à nos aînés. Les solutions proposées par les équipes finalistes de cette édition du concours vont certainement y contribuer dans le moyen et le long terme. Je vous en remercie. En novembre 2018, le premier ministre François Legault confirmait la création de zones d'innovation et me confiait le mandat de déployer ce projet de grande envergure. Le déploiement de ces zones d'innovation vise l'ambitieux objectif d'accélérer le passage dans l'idée au marché. Malgré la situation actuelle causée par la pandémie de COVID-19, ce projet n'est pas mis en veilleuse, loin de là. Ça reste au cœur de nos préoccupations stratégiques. Je rappelle que les zones d'innovation seront des territoires géographiques délimités où seront regroupés des acteurs de l'enseignement, de la recherche, de l'innovation, de l'industrie et de l'entrepreneuriat. In closing, I want to wish good luck to all participants today. I'm inviting you to keep prioritizing our collaborations and innovative partnerships. Organizations of all sizes, businesses, research centers, investors, everyone has a role in creating more innovation and competitive environments in Quebec. Merci. Thank you very much.
Merci beaucoup, Monsieur le ministre. Je pense que maintenant, c'est à Dr. Kevin Lachapelle qu'on passe la parole. Kevin?
Bonjour tout le monde, and welcome. It's my pleasure to welcome you to the McGill Clinical Innovation, and this is the third year. I'm Kevin Lachapelle. I'm professor of surgery, and I'm the chair of surgical education. I had the opportunity, along with one of my colleagues, Dr. Jake Barrelette, and the support of department chair at the time, Dr. Freed, and Dr. David Eidelman, to create an innovation program at McGill. And what you're seeing now is, I think, part of the fruits of that labor. I want to send out a special thank you to Dr. Jake Barrelette, who is not the host this year. He's given that work to me, which I have graciously accepted, so you can all see my face instead of his. So I'd like to give a shout out to Jake Barrelette, who's watching this on streaming. So this event really is our approach at McGill, I think, to try to demonstrate how much real innovation is being done at McGill from the bench and to the bedside and into commercialization. And it's also meant to stimulate people. And I think from what I've heard and what I've seen, the groups and the competition has become much more intense, and the quality of the innovations and the quality of the presentations has increased dramatically. So just bear with us, though, during this kind of video technological innovation as well. One thing I do want to try to stress, especially with the presenters, certainly those that are going to be presenting for the competition, that you have 8 minutes and 5 minutes for questions. And we are going to have a hard stop on that. So when you see my hand go up, that means you have 2 minutes left. All right? And after 2 minutes, it's a hard stop. We're going to really pull the plug on you. Same thing for 5 minutes, or else we're never going to finish. So that being said, I'd like to introduce Dr. HAKIM, who was actually the innovator of this prize and of this day. And I'd like him to say a few words to the group.
Thank you very much, Kevin, and thank you very much, Dean Eidelman, for your welcoming remarks. They are very much appreciated. Je désire aussi dire un grand merci au ministre de l'Économie et de l'Innovation, l'honorable Pierre Fitzgibbon. Nous sommes très honorés de votre présence et encore plus de votre intérêt à connaître les détails de ce programme qui a été conçu afin d'encourager les membres de la communauté facultaire à McGill à concevoir et à développer des idées innovatrices qui auront un impact direct et durable sur les soins de santé au Québec, au Canada et même à l'échelle mondiale. Ceci prend place pour la 3e année, mais malheureusement cette année, nous ne sommes pas face à face, alors que nous faisons face aux défis exceptionnels que pose la pandémie COVID-19. The idea behind establishing this clinical innovation competition came about while I was looking at a number of startups and developing healthcare companies in the United States. Somewhat to my surprise, many of the key principals in these companies were originally from Canada, and a few listed McGill in their education background. And I thought, how come these individuals are not developing these companies in Canada? The topic came up in a family get-together. And as some of you may know, my family has many connections to McGill. My brother, Antoine Hakim, is a member of the Canadian Medical Hall of Fame and a recipient of the Gairdner-Whitman Award, and was a neurologist at the Montreal Neuro Institute for many years. And my sister and a couple of my nieces and nephews are graduates of McGill. And I'm proud to say that I'm a graduate of McGill Medical School and was a resident in medicine at the Royal Vic. If anybody's interested, I can show you my scars. So at the family get-together, we decided to establish the HAKIM Family Innovation Competition, which is in its 3rd year and successful beyond our wildest imagination. I'm very pleased that this year we had 46 applicants. And after Jake Barolette prioritized the applications, the judges, including myself, ended up with 15 proposals to review. And I must say, as I read the 15 proposals, I kept thinking to myself, why can't we have 15 prizes? Because each one of these applications or proposals came up with a new idea that, if successful, would have a direct positive impact on healthcare in Quebec, Canada, and globally. And to prove the point, Sarah Stack, one of the event officers, has interviewed last year's winners of both awards and published those brief interviews in McGill Med E-News recently, and she'll send you the link to those. It was very heartwarming to read about all the progress made by these prize recipients in the last year and their acknowledgement of the impact that this competition and these awards made in the progress of their project. With that brief background, I'm pleased and honored to turn this over to Dr. Kevin LaChapelle and start the serious part of this meeting.
Thank you, Dr. Hakim. And so now we're going to get into some of the nitty-gritty. First off, I'd like to introduce Dr. Marcel Baer, who's co-director of McGill Interdisciplinary Initiative in Infection Immunity, MI4, and the director of McGill Infectious Disease. And as a director of infectious disease amid a COVID-19 pandemic, you can imagine that he is probably the busiest person, at least at this university anyway. And he's been the source of many questions and comments, and certainly a lot of interesting ideas. So Marcel, I'll let you give the presentation for the MI4 prize.
So thank you, Kevin. Thank you, Dean Eidelman, Monsieur Fitzgibbon, Dr. Hakim. Hello, virtual colleagues, virtual judges, and virtual innovators. I would just like to ask the slide to be advanced because you don't really want to know about me. So next slide, please. So there's going to be an MI4 Innovation Prize that I'm introducing. And if you go to the next slide, I will tell you first who's MI4, and then I'll tell you who has earned the prize this year. So MI4 is our cool-sounding acronym for the McGill Interdisciplinary Initiative in Infection and Immunity, because that takes too long to say. And here's a picture of the three directors, Don Shepard on the left and myself top right. That picture was taken when Don and I rounded on the COVID ward about 3 weeks ago, and he was my COVID ward resident that day. And Marie Hudson actually works at the Jewish General, so believe it or not, she was photoshopped onto the image. It probably looks like it, and Marie is on the line, but that's at least the three of us. And since we're working virtually, I think we're all allowed to have some liberties with Photoshop. So what is MI4? We are a collection, a rassemblement of over 200 world-class investigators with diverse expertise in infection and immunity research. And these investigators are at the Jewish General, at the Research Institute, at McGill University, at McDonald campus, at the Neuro. Basically anywhere people is doing infection immunity research who's McGill affiliated, McGill associated, we are finding ways to bring these people together in ways we hadn't imagined before, and we continue to do so despite COVID and despite Zoom meetings. So what do we seek to accomplish? We seek to accelerate the pace of research and innovation. So that's where we are firmly aligned with the CLIC competition, because one of our deliverables will be real-world solutions for grand challenges in infection and immunity. And how do we do that? To date, we have been very fortunate to receive generous gifts, such as the one from the Doggone Foundation that's going to this particular competition. And through that, we've had competitions for incubator grants, for open access technology platforms and co-funding opportunities. To date, we have committed over 10 million, and this is in just over 2 years, to 19 incubator grants that we call seed fund grants, and to 48 emergency COVID-19 grants that we were able to put through in the past 3 months in a series of competitions. We fund 5 open access technology platforms, including a level 3 hot zone where we can cultivate SARS-CoV-2 and do experiments with the pathogen causing the pandemic. And we work in partnership with people on campus. And today, of course, the partnership is with CLIC. This year, we are pleased to announce that the CLIC MI4 Innovation Prize of 2020, and I am now giving a virtual handshake because I'm not able to welcome you, goes to 4 students in bioengineering: Alexandra Bevacqua, Ali Najmalden, Domenico Lopez, and Jiachi Wu. So congratulations for your proposal, which is called Minutes to MRSA, a microfluidic-based diagnosis of methicillin-resistant Staph aureus. Congratulations to all of you, and I wish I could shake your hand in person. And it's very exciting to hear that our students are already putting applications together and writing business cases for why their microfluidic device could revolutionize the management of antimicrobial resistance. So thank you, and I'll pass it back to Kevin.
Thank you very much, Marcel, and again, congratulations to the winners.
I'm Alexander from Minutes to MRSA, and we are developing a technology which will enable clinicians and nurses to screen hospital patients for MRSA within minutes. We aim to disrupt the MRSA testing market by developing a radically faster, easier to use, and affordable diagnostic test. MRSA is a life-threatening bacterial infection. It is a multi-drug resistant superbug. It can spread through indirect contact, and it is asymptomatic for up to 10 days. Let's set the scene up with a story. So Time reporter Chris Linneman underwent a standard surgery after tearing an anterior ligament in his left knee. Now the surgery went well until one morning when he bent down to find his knee had swollen to the size of a melon. The doctors found out that he had a MRSA infection and had to be operated on. Now after that surgery, he was sent home, but he had to be readmitted again, this time with a swollen face. Thankfully, a spinal tap indicated that the superbug did not spread to his brain yet. After multiple surgeries and a bill of $300,000 US, Mr. Linneman was able to fully recover. The story of Mr. Linneman is a reminder of how costly and potentially fatal bacterial infections can be if undetected immediately. MRSA infections cost American hospitals $7.2 billion US annually. It causes 120,000 infections annually, and even with medical intervention, 20,000 people still die annually. We introduce MRSagent. It's this small diagnostic device. In fact, we're going to put the entire lab right in the palm of your hand at the patient's bedside. All of the lab protocols for MRSA screening are miniaturized into the chip, effectively automating all of the labor and enabling the screening to be done at the point of care. Now, Medica will dive into the market opportunity of our product and our progress.
Thank you, Alexander. MRSagent is radically different compared to the other options currently available on the market. The current standards include bacterial culture and PCR amplification, which take over 24 hours and 8 hours respectively. The alternatives on the market form two classes of potential competitors, a reusable kit method and a multiplex method of scanning for MRSA. Both take 5 hours, are expensive, have complicated upkeep, and sensitivity is lower than the traditional test. MRSagent does the screening in minutes, is easy to use, has a sensitivity matching the current standard, and a lower cost for the customer. As an early-stage diagnostics engineering startup company, our goal is to get people excited about our innovation and prototype our design. The MI4 Innovation Prize will enable us to build our device and speak with experts in business and medicine. The greatest asset of our startup is our team. All of us are trained to have a very interdisciplinary mindset as part of the bioengineering program at McGill, with half of us specializing in high-throughput screening and the other half in biomolecular engineering. Thanks.
Great, thank you for that presentation.
Now, after this part, we're going to go into the final team pitches. And again, we're going to go in a sequential fashion. So first, I'd like to describe what we're actually competing for. We're competing for money, that's for sure. We're competing for prize money, but we're competing for two different types of prizes. So one is the HAKIM prize, under which this competition is named. It's $10,000 and with up to $50,000 of in-kind support. And really this is given to the winners where the innovative device has the most potential for commercialization. So that's one aspect of this component. The second aspect is the Roy Prize, and really this one is about the best solution for an unmet need. So there's a little bit of a difference in terms of the prizes and what their proposals are. This one is also a cash prize of up to $9,000 with up to $40,000 of in-kind support as well. So both fantastic prizes. I think we've had previous winners that have been very successful, and you can read it again, as Dr. Hakim said, in the McGill e-news. But this has been a very coveted prize, I can say, and there's been a lot of good press and good competition with it. So again, we're competing for 2 prizes, as I mentioned earlier, and it's my pleasure to introduce GyroClear, who will be presented by Aidan Reich.
Hi, thank you, Dr. Lachapelle. So today I'll be introducing GyroClear to you. So you might be wondering why I'm starting with a Formula One racing car during a clinical innovation pitch. Well, I'll only explain why shortly. For now, I'll introduce to you the observation we had while spending a few weeks at the Montreal General Hospital. We noticed a recurring problem that happens during laparoscopic procedures. So as you can see here in this clip, the camera is slightly blurry, and so the surgeon has to remove it from the body, clean it with a sponge or a towel. Upon re-entering it into the body, you'll notice, due to temperature differences, that the camera will fog up, as we can see here. And so once again, the surgeon will have to remove it from the body, clean it down with a sponge, submerge it into some warm water, and all this results in a prolonged duration of the operation, increased risks of infection, a disruption of the surgical team flow, and overall a frustrated surgeon. And so to put all of this into perspective, in 2019, over 4.8 million laparoscopic procedures were performed in the US alone. When we spoke to surgeons, 13 out of 14 of them shared the same opinion as us and wanted to find a solution to this problem. And so as promised, I'll come back to the F1, where it's because F1 also encountered a similar problem as the one in laparoscopic surgery. So if you were to watch a race on TV during an overcast day a few years ago, you might have been underwhelmed by this image with a lot of raindrops on the lens. And so F1, having some of the greatest engineers and constantly pushing the boundaries of innovation, developed this device that encapsulates the camera and has a spinning lens, allowing for constant clear images despite the rain. As we can see, in the 2018 Brazilian Grand Prix, we had a really crisp image despite all the rain they suffered from that day. So why not inspire ourselves from these F1 technologies? That's what we did at GyroClear. Our novel device aims to set a new standard for minimally invasive intra-abdominal and thoracic surgeries with its innovative design. As we can see here, it's a protective sleeve that would slide onto a laparoscope or rigid endoscope. And as we zoom in at the tip of it, we can see that the lens would be spinning, creating centrifugal force, allowing to instantaneously remove debris such as blood or tissues. It will also generate an airflow that would act as a barrier against smoke particulates due to the cauterization of tissues within the body. And so the benefits of this are, firstly, that we would allow constant clear visibility. A study has shown that surgeons lack proper visibility 43% of the time. And so our device would deliver that constant clear visibility 100% of the time, allowing surgeons to operate safely around critical anatomical structures. And so we'd be able to increase patient outcomes as well as decrease the frustration of the surgeon. Second, we can shorten the length of the surgery by around 5 minutes per hour of operation. This reduces the unnecessary time variable costs, such as overtime, allowing hospitals to save up to $300,000 per year. Finally, we could completely eliminate the need of removing the camera from the body to be cleaned, and therefore we could reduce the potential risks of infection, as well as once again decrease the frustration for the surgeon and the surgical team. And it's for these reasons that, much like the 13 surgeons we've spoken to, will be demanding for our device. One might expect some competitors out there. The competitors in this field fail to address all the required features of a good device. However, with our unique and inventive design, GyroClear meets each and every one of these criteria. Plus, we have some added benefits of constant clear visibility, as I've previously mentioned. The competitors only clean their lens once it's already been dirtied, similarly to a windshield wiper. We also have a reusable device, whereas our competitors are disposable, and therefore we're more cost-effective and environmentally friendly. To give a little idea of the market potential, we have this equation that outlines bottom-up analysis of the market. And so if we start on the left, we can see that there's 1,700 hospitals and ambulatory surgery centers doing these types of procedures. they're each doing around 16 procedures per day and therefore would need 16 of our devices. These devices would have to be ordered in between 3 to 4 times per year, considering that our device is reusable, but up to 100 operations, similarly to a laparoscope. Therefore, they have to order it 3 to 4 times per year. Finally, our device would cost around $1,250. So considering all of this, the US market comes out to $119 million per year. When we take that and multiply it by the globalization ratio of 3, we can transfer the US market to a more global market, which allowed us to estimate it to $357 million per year. We've also devised a go-to-market strategy where first we identified our beachhead market, which are general surgery hospitals performing thoracic and bariatric surgeries, because these are considered to be very messy due to the cauterization and cutting of tissues. And so phase 1 of our strategy involves marketing our device through medtech conferences, exhibition halls, and through our KOLs, distributing the device through our medical device distributors. And as our company grows and starts to expand to a more international market, we would then enter phase 2 of our strategy, which would be to partner up with an OEM, allowing us to integrate our device into an actual existing laparoscope. And so this innovative path shows some of the milestones that we're going to be starting within this year. As we can see here, we went through the surgical innovation program that allowed us to find a problem and a need, as well as the idea for it. We started the research and development of the product, and it will be a process going through all the way through the end of the project. Sometime during the summer, we will do our patent application, which will allow us to claim that really important priority date. And then we'll enter with the regulatory affairs, setting up our QMS, registering for our ISO 13485, as well as our establishment and listings with the FDA. And we'll go through a 510K process as a class 2 device. By the end of 2020, we want our initial alpha prototype ready for testing, where we would do our preliminary in vivo trials shortly after in animal models, continuing through 2021. And so to do all of this, we have a very diverse team. Pierre-Paul and myself have backgrounds in engineering and have the technical skills to create a proper prototype. Dr. Fried and Kenny are acting as consultants, helping us out with the clinical trials and in vivo trials, and they're our healthcare specialists. And finally, we have Sridhar, who's our business specialist and has over 15 years experience as a project manager and will help us keep on track with this project. And so if we win the CLIC competition, we use the prize money to help fund our patent process, as well as all the other services included in the prize to help develop our product to the next level. And so right now, I'd like to thank you very much for your attention and open up the floor to any questions. Thank you.
Thank you, Diane. Thank you, Aidan, for the excellent presentation. A little puzzled with your commercialization strategy. Why would you not go to a partner with an OEM or an endoscope manufacturer from the very beginning?
Well, we thought that if by selling our devices through medical device distributors, we would get more traction and recognition, and then it'll help us go through an OEM. Obviously, if we do get a patent fairly early on, we might go directly to them and try to sell the patent directly and talk about integrating it directly to their device. This would actually help us create a thinner device because it'll be directly integrated to the laparoscope and therefore has even more benefits. So it all depends on how early we get our patent and how things go on. But we are considering possibly going that direction.
Thank you.
Great, thank you. So next I'm going to call on Stephen Arliss, please.
Yeah, thank you. Thank you for your presentation. Oh, thank you. And I just have a question as well about your business model. I think the product seems to address a significant unmet need, so I congratulate you on that. But I'm just wondering about your business model of having a reusable device in this day and age where more and more of the world is moving towards single-use disposables. What made you go this route as opposed to a fully consumable disposable model?
Well, speaking to surgeons, a lot of them were interested by the reusable device as opposed to the disposable one. Obviously, this was for reasons such as being environmentally friendly. Also, when we discuss costs, because we would be able to amortize the cost through many operations, this device would come out a lot cheaper than other ones. Competitors come out as low as $200 per operation, whereas us, we're able to come out around $70 per operation, which would cost a lot less for the hospitals as well. And that's the reasons why.
Thank you.
Okay, so next we have Dr. Hakim.
Thank you for a great presentation. Sorry, am I muted?
No.
No.
Okay, great. I wanted to find out whether you have any in vivo models that you've used this new device at. Have you done it in vivo yet?
We haven't done it in vivo yet. Unfortunately, we kind of got pushed back due to COVID. We weren't able to get as far as we wanted with the development of our product, and therefore couldn't get our final prototype ready. As I shown in our sort of timeline, we want to have a final prototype ready by the end of 2020 and do our in vivo trials around that time. But we'll be ready to do them, like I said, around 2020.
Because you probably may be able to do it in animal studies.
Definitely. We're thinking of doing the preliminary ones in vivo. This is something we talked in animal models. We've discussed this with Dr. Fried, and he agrees upon it. It would be a lot less expensive at first to go through that model. And therefore, that's what we're planning on doing.
Okay, thank you.
Thank you.
Great, thank you. Can I just ask you to stop sharing the screen, please? And then we'll continue with Carmela DeLuca.
I think my screen is not sharing anymore. Is that possible?
Okay, perfect. All right, my mistake. Thank you.
Thank you.
Quick question. Carmel.
Yes. Hi. I just wanted to give a comment too, since I know you've talked a little bit about the solution and it's sort of related to sort of some prior art solutions, although in a different context, and that you're not planning to submit your patent until sometime this summer. And I know money is always an issue for these kinds of things, but you should really keep, if you've been discussing the solution and presenting on it, you really want to make sure that you're at least 1 year, if you're not able to file before, if you haven't been able to file before, that you really want to file within 1 year from the first time you've disclosed, so that you get broad protection. Because if the pieces are can be determined from what you've described, taking what's already known in the prior art, you're going to be limited in sort of what patent protection you'll be able to get. So you want to move on that quickly.
Yeah, I agree. We actually spoke to a patent agent, and we made sure that we're really not disclosing anything, any of the structures that could be any problems, causes problems with that. We're really keeping it high level, obviously not talking about specific structures or mechanisms or how it will be functioning exactly. And it is quite different from what F1 already has done as a prior art.
Okay, good.
Thank you.
My hand up doesn't work, so maybe you could let me know how I can signal to you.
Okay, maybe you can do a thumbs up instead. Can you do that? If you go on reactions on the bottom? Or yes. You can just physically raise your hand and I'll look for that. So we'll call on Brent Norton, and then after that, you, Nadine. All right, so go ahead, Brent.
A bit of a follow-up to Stephen's question. In the image you showed of your sleeve, at the very sort of, the distal end, the part that's inserted in the body, there looked like moving parts. So really 2 questions. Were there moving parts? And if there are, was there any commentary from Dr. Fried or other surgeons about having moving parts deep inside the body? And secondly, around the sterilization for the reuse given the moving parts?
Yeah, so these are things we've considered. We've spoken with the surgeons. We've also looked at, depending on regulatory affairs, obviously to get approved by the FDA, they won't approve something that could harm the patient. And therefore, we were looking at it, and through ISO 13485, there's specific tests we can do to make sure that the spinning motion, the actual part that spins at the end, won't cause any harm to the patient. So far, we've done some testing on a bench model. It won't spin fast enough to create any harm. Obviously, we'll do more elaborate testing and keep it tracked in a DHF and all. But that's to answer the first question. The second question, could you please repeat that, please?
Yeah, regarding sterilization of the instrument given moving parts, and obviously there'd be parts that are behind other parts.
Yeah, so for the sterilization, It's something we're considering, obviously, because it's reusable. It will be to be tested and determined depending on the final parts that we have created. Obviously, with the research and development, it's a continuous process of improvements, and therefore some parts will change. We don't have exactly the final model yet tested for the parts. However, we're making sure that we're going to be using materials that could withstand high temperatures through autoclave, as well as other products, other pieces that could withstand that. We're really trying to make sure that would work out.
Thank you. I'm very sorry, but unfortunately we've reached the end of our time and we have to move on to the next team. So I'll invite PLAKK to come and take the stage, please.
Start.
Yeah, just go ahead.
Okay, thank you very much. Certainly, we're all familiar with stroke and its devastating consequences, either as a patient, family, friend, or doctor. But let's talk about the problem that lies beneath and causes strokes. The real problem is atherosclerosis in the arteries, also known as a PLAKK. It is a leading cause of death worldwide, and it is very costly to the healthcare system. Yet, there is currently no accurate method to predict if this is going to affect us. So what is the current practice to diagnose and manage these plaques in order to prevent strokes? Typically, a patient comes in either after having a stroke or without any symptoms. Ultrasound is used routinely to assess solely the degree of blockage caused by the plaque in the carotid arteries of the neck that feed blood to the brain. And when the blockage is high enough, surgical intervention is recommended by guidelines. However, clinical practice leaves us with limitations. Blockage is a poor determinant of stroke risk, leading to high clinical uncertainty. Approximately 40% of plaques causing high blockage will never cause a stroke, while other plaques with lower blockage can cause strokes. There is unjustified patient risk, as some patients are offered unnecessary surgeries, while others with high stroke risk are not receiving proper treatment. Lastly, there is unnecessary waste of resources due to inappropriate treatment allocation. So how can we address these limitations? The truth is, plaques have complex composition, which is a better indicator of stroke risk than blockage. The presence or extent of certain features within the plaque dictate the risk of a plaque rupturing and causing a stroke. Following surgical removal of the plaque, we can characterize it under the microscope histologically, which is the current gold standard method. However, to date, we do not have any clinical tool to assess plaque composition before the plaque is removed. PLAKK addresses this clinical need with the creation of an artificial intelligence-based platform to revolutionize the prediction, treatment, and prevention of stroke by providing a comprehensive characterization of atherosclerotic plaques. The scientific foundation of PLAKK started 20 years ago. My journey started with using simple computer-aided methods on ultrasound to classify high-risk plaques, and later on with more complex digital image analysis. Since 2009, I have created a growing biobank of paired carotid plaque specimens and ultrasound images that allowed me to identify specific ultrasound plaque features that correspond to histological plaque features. This past work feeds into our current innovation, which will take advantage of recent advances in AI. This is possible given our unique biobank of data obtained from approximately 600 patients who have undergone surgical removal of their plaque, making it the largest biobank of its kind, at least in North America. From these patients, we have collected ultrasound plaque images preoperatively and plaque specimens post-surgery for histological analysis. Using these paired images, we will validate our ultrasound findings with the gold standard histology and develop our AI algorithms using convolutional neural networks. So the algorithms will predict from ultrasound alone how the plaque and its individual features would appear histologically without having to remove the plaque. So how will our platform work? An ultrasound image is uploaded and analyzed, and the final report is generated with a quantification of plaque features and an overall stroke risk score. How will our innovation change clinical practice? Firstly, it will improve the accuracy of a patient's stroke risk by 2.6-fold compared to blockage alone. Secondly, it will improve clinical decision-making, including decision for surgical intervention. Lastly, we can save the healthcare system more than 1.2 billion in the US alone by reducing the number of unnecessary surgeries. This does not even account for the savings associated with preventing a stroke. And we have the right team to achieve product market fit. Together, we have diverse expertise in medical science, business strategy and commercialization, and 18 years of AI experience. And we are supported by a strong and diverse advisory board and mentors. There are few competitors in the field. Zebra Medical Vision and Caristo both use AI in coronary CT angiography to characterize only one plaque feature each in the arteries of the heart. This is a major limitation, as cardiovascular risk is driven by a combination of plaque features. LifeQ has a similar mission to ours using ultrasound images. However, they do not use AI technology, and thus their analysis is less accurate and more time-consuming. Most importantly, none of these competitors use the gold standard histology to validate their imaging findings. PLAKK sustainable advantage is our proprietary biobank and my 20 years of experience in this field. For this, we're in the process of filing a background IP with McGill, which will then be licensed to PLAKK. Our foreground IP will utilize a layered patent strategy with 3 parts. The actual image transformations will be patented. The AI algorithms will be kept as a trade secret to ensure others will not repurpose them. The process of applying these algorithms to each plaque feature and the overall risk score will be kept as a know-how. Worldwide, 1.5 ultrasound scans for plaques are performed yearly. Of those, 300 million are carotid scans. Even if our platform is only used for more uncertain patient cases, this will provide an estimated serviceable obtainable market of 180 million scans yearly. We have 4 arms to our commercialization strategy. Through our global collaboration, we will first enter the research market, since it is more likely to adopt new technology and to begin generating initial revenue, as they will be our first customers. Our strategy is then to enter the US clinical market first and obtain FDA approval as a Class 2 medical device by completing the 510(k) path using a predicate device. We're evaluating possible market entry strategies, going in alone or partnering with large ultrasound manufacturers. As a main strategy, we will integrate our platform into already existing technology through partnering. We'll use software as a service model and charge a fee of $1 per scan, projecting a potential yearly revenue of 90 million, assuming 50% market penetration. At present, we aim to finance PLAKK on non-dilutive funding sources. If need arises, we will seek seed funding in consultation with our advisory board. This will help us complete at least 5 PLAKK features per year, file for provisional patents, test our MVP, and begin our prospective clinical studies. We intend to start filing for FDA approval within 5 years.
1 minute, Stella.
Eminently, our platform has the potential to be adopted to several other markets. Our platform can be extended from carotid atherosclerosis to be applied to plaques in the heart and in the legs, and broadened to other imaging modalities such as MR angiography and CT angiography. Most importantly, we can aid research and clinical practice by monitoring the effect of drugs such as statin directly on plaque composition. So choose PLAKK because our technology will empower clinicians to revolutionize decision-making and clinical care hand in hand in order to predict, treat, and prevent strokes. Thank you.
Thank you very much, Stella. Very nice presentation, exciting work. Diane, would you like to ask the judges for questions, please?
Yes. I see that Steve Arliss has a question, so please go ahead, Steve.
Field for a long time, and this could be a game changer. So congratulations.
Thank you very much.
I've dealt a lot in the world of vulnerable plaques versus stable plaques. Could you care to comment on the ability of your technology to differentiate between vulnerable plaques and stable plaques, which is really the nemesis of a lot of interventional cardiology right now?
This is a great question. This is the work of my life. So this is the key, that we have to differentiate between the plaques that are at high risk to rupture and cause strokes and plaques that are really stable and they will not. So there are several attempts. People have used ultrasound, have used CT, you have used MRI. Yeah, CT angiography, MR angiography, contrast enhanced MR. The problem is that all of these different efforts have been actually still at the stage of research. There's a lot of research, although most of them are looking at one feature per one feature at the time. But the truth is that they haven't reached the clinical practice. And one of the reasons is that, these other more sophisticated modalities, like the CT or the MRA, they're actually more expensive. Sometimes there's radiation, and they're not widely used. So the purpose of using ultrasound is to really maximize what is out there and what is done anyways in clinical practice. So everybody who has a plaque or who is supposed to have a surgery will have an ultrasound anyway. So we get some information and we want to maximize this information. So we get it really in the clinical practice and not in research settings or
So Stella, my basic question is, if you can differentiate vulnerable plaques from stable plaques, that just give me a yes or no, because if you can, it's out of the park home run.
Yeah, that's exactly that, because we were looking at the 10 different features, histological features that actually render the plaque stable or unstable, and we actually can say which features exist in each plaque, and therefore give an overall score based on the individual features, if this is a plaque is going to rupture or not.
Thank you.
Absolutely that.
Great, thank you. So now we'll go to Dan Roden next, please.
So I want to press on that issue a little bit more. It seems to me that in order to define a plaque as stable or unstable, you have to image it, and then you have to watch it and decide whether it's going to rupture and create an obstruction, or whether it's going to sit there, regardless of whether it's a 70 or 80% occlusion. So I'm going to actually sort of show my face even. Short of that, I think what you have is a tool that will allow you to look at an ultrasound image and parse in great detail whether there's a fibrous cap, how big the lipid core is, but the extrapolation to stability or instability, I didn't see it in the write-up, and I didn't sort of feel compelled by your answer to Steve. So can you sort of, are there data that say I've imaged a bunch of plaques, and now I followed them for 3 years, and here's the ones that ruptured and here's the ones that didn't rupture?
Right.
Okay.
Right.
Okay. So in fact, this tool that we are creating is a diagnostic or decision-making tool as a first phase. So I want to see now how the plaque, I look at the plaque on the ultrasound, and I want to predict if I were to take this plaque out of the patient, how it would look. This is the idea. So I want to see now your risk. In a second phase, this will work as a preventive tool and as a therapeutic tool, in the sense that we can monitor the plaques, we can see how they can evolve, and we can see how treatments actually change the plaque. But at this moment, we want to make sure that when I do an ultrasound, I can predict or I can mirror how the histology would look like. So in other words, I can see if a patient has 90% stenosis or blockage, and then yet he has very stable plaque according to the features, and then in that case my decision as a surgeon might change. Okay, I have a stable plaque, I don't really need to operate on this.
There are lots of other questions. I just want to say that it'll be hard to convince surgeons to not operate on a 90% stenosis.
Yes, but I can convince them also to operate 20% of people who are missed there. So patients who actually have unstable plaques, but they don't have the stenosis criteria to actually operate on them. So it could be a reshuffling actually of operating on the right patient. So it's one thing is to operate, but the second thing is, as you said, it's to monitor and also to assess how the drug work directly on the plaque, and not, like just for a statin, for example, reducing your cholesterol, but how it changes your plaque.
Thank you, Stella. We just have just a few seconds left, so we'll let Dr. Hakim ask his question, and then we'll have to move on to the next. Yeah. Thank you.
Thank you.
Are you primarily depending on analyzing ultrasound data, or do you have the ability to analyze CT scans, MRIs, and so on, which are often used in that? That's one question. And quickly, can you identify calcifications in these plaques?
Yeah. So I'll start with the second question. Absolutely, calcification and all the other features that render the plaque unstable, or stable or unstable. And the first question is yes about CT. So we start with ultrasound, as I said before, because it's very widely used and it's already the clinical tool. And by recommendation, it's the first test that we do. And maybe we just rely on this to go for surgery or any other medical treatment. That was the idea. So we basically our denominator is much greater if we go with ultrasound that is used anyways in clinical practice at this point. In other words, maximize what we use from ultrasound. But nothing stops us from actually using this technology. And this is, as I said in my second to last slide, to go actually to other imaging modalities, including CT angiography and MR angiography. But we first want to start with what is more widely used to increase our impact in the market and in the and in terms of clinical care.
Thank you.
Thank you very much, Stella. That was a great question and answer period. So we'll move on to the next team now. We'll ask Jeremy Levitt of Stenoa to take the stage, please.
By the time it takes me to complete the next 3 sentences, which is approximately 40 seconds, it's estimated that someone, somewhere will suffer a cardiac arrest. Stenoa aims to work live, in real time, that is less than 1 second, to maximize the positive outcome of a life-saving intervention. Good afternoon, ladies and gentlemen. My name is Jeremy Levitt, and I'm the founder and CEO of Stenoa. And today, I want to speak to you about 3 things. There's a problem in the way we currently do things, causing more people to die than necessary. Stenoa is a novel approach that addresses an unmet and an urgent clinical need at a fraction of the cost. And most importantly, I want to speak to you about the future. Because by the time I finish this presentation, I hope to convince you that Stenoa isn't just a solution to a need. Stenoa is disrupting an industry. You see, coronary artery disease is a leading cause of death worldwide, and it's claimed more than 9.5 million lives annually. What you're seeing in this video is a visual angiogram from a cardiac catheterization of a patient with coronary artery disease. The coronary arteries are opacified by contrast injection, and operators anatomically assess the vessel anatomy for potential blockages. The challenge which interventional cardiologists face is that visual angiography can only assess the anatomical severity of the lesion, and it's a subjective assessment based on physician experience. This provides an incomplete picture of the amount of blood perfusing the heart, which is a true physiological standard that should guide treatment, including stent placement and need for open heart surgery. Flow rate doesn't necessarily correlate with anatomical severity, and fractional flow reserve addresses this by using an invasive pressure wire, which is inserted and guided to the lesion, measuring pressure before and after stenosis. The more flow limiting the lesion, the lower the FFR, with 0.8 used as a cutoff. There is really robust data that suggests how effective FFR is. Routine FFR reduces death or other cardiac events by 34%, changes the management strategy of patients in 43% of cases, and is cost-saving in 91% of cases, identifying the impact which precision guidance can have on the healthcare system. Bottom line is that routine use of FFR saves lives and is guideline recommended. But while it saves lives, it has many drawbacks. Invasive wires increase patient risks, they contribute to additional procedural time, and they come with substantial costs. Despite long-term use showing improved outcomes with FFR-guided decision-making, its use remains significantly underutilized in practice, with FFR being used in only 6.1% of interventions for intermediate coronary lesions, exemplifying the limited uptake of this technology. Ultimately, patients are dying because the standard of care is not being routinely implemented, and treatment decisions, such as stent placement, are being made with limited objective support, sometimes causing stents to be unnecessarily or wrongly placed, or on the other hand, causing patients not to have stents placed or not to be sent for surgery. This is a problem. So what if we can measure FFR, the true functional and ischemic severity of a lesion, on every artery, without even having to use the wire? Stenoa was born to enable operators to routinely perform FFR measurements, although with Stenoa's live, virtual, wire-free FFR. Stenoa proposes to automatize the FFR evaluation using machine learning algorithms. Stenoa is the first to enforce dynamical consistency in coronary artery segmentation, ensuring angiograms are interpreted comprehensively and not just analyzed at the single frame. This provides interventional cardiologists access to live, real-time information for intraoperative decision-making. And finally, Stenoa results in improved patient outcomes, safety, and cath lab workflow. Stenoa's solution isn't cost prohibitive and doesn't introduce any additional patient risks. There's a need to assist operators in making quantitative decisions, and Stenoa addresses this by providing quantitative measurements in a real-time, convenient, and easily accessible manner. Essentially, Stenoa promises to improve healthcare outcomes by democratizing the ischemia-driven revascularization process. The Stenoa approach relies on big data. In working with the McGill University Health Center, we developed a proprietary hierarchical database structure where we extracted patient-level angiographic data from hospital picture archiving and communication systems and linked it to the patient's clinical course through their electronic health record. In compliance with standard operating procedures and regulatory requirements, we created a unique database that is HIPAA compliant and anonymized with an internal dictionary. The beauty of our work is that we have anonymized, yet patient-linked procedures, which are 10 to 12 years apart in some cases. This is historical data with procedural tracking that has infinite potential. It's a novelty in its own respect. Taking the time to organize the data this way has been a major contributor in allowing us to get to where we are today. This is what a normal coronary angiogram looks like without any Stenoa processing. Our technique deploys a series of automated machine learning algorithms, which we visualize for you here, whereby we separate the layers of this 2D angiographic video, extract vessel features, perform motion-consistent frame interpolation, and implement an active surfaces approach, resulting in pixel-level, feature-based, dynamically consistent segmentation. A first for coronary angiograms. We not only segment the edges, but the vessel opacifications as well, allowing us to compute density gradients and look at coronary flow from a physiological perspective. Our algorithm doesn't just do this on one coronary angiogram. The machine learning software is capable of performing this segmentation under wide anatomical variability, which we validated with the right coronary tree. Our algorithm not only detects the edges of the coronary arteries, but their actual interior opacities and density gradients. This will allow us to, based on the regions of interest, predict and assign virtual FFR values. Now let me tell you why we believe that Stenoa works. To ensure the quality of our algorithm, we performed automated segmentation in 2 different pipelines and validated our segmentation results on the right coronary tree. When compared against other general segmentation models, we were pleasantly surprised to see Stenoa perform extremely well. Stenoa has a high sensitivity due to having very few false negatives, and this informs us that we are accurately segmenting the local dynamics of coronary edges, an important measurement we can be confident in. So we began this path in 2018, where our product was conceived and developed. From there, we worked with the MUHC's Review Ethics Board and Contracts Office to access large data and organize our own respective database where we train our algorithms. We then proceeded with retrospective internal validation and are currently pursuing that, and are now at the point where we need the support of the clinical innovation competition in order to proceed with intellectual property protection. We are incredibly excited to follow this path, which will allow us to work with collaborators in externally validating our algorithms, proceeding with inhuman prospective studies, a randomized control trial, and eventually commercialization. The real barrier to competitor entry is not the patent. It's the proprietary database architecture from which our algorithms are built that I spoke to you about earlier in the presentation. Nevertheless, Stenoa's algorithms constitute statutory subject matter which is novel, inventive, and has utility, which will allow us to file for provisional international patent protection. I would just like to briefly highlight the ways by which regulatory requirements can or have been adjusted by our project. We received REB approval and collaborated with the MUHC to access data. We HIPAA compliantly anonymized our data and proceeded with 2-factor authentication cloud storage solutions, and we acquired the Stenoa domain name. With the support of the clinical innovation competition, we hope to incorporate and trademark this product, protect its intellectual property, proceed with CE, 510K FDA, and Health Canada approval. Now let me address commercialization briefly. We hope to implement a continued cash flow licensing model where we work with software providers to develop a one-time implementation fee and pricing that's proportional to volume, followed by a targeted evidence-based marketing campaign to attract licensing partners worldwide, with our first large conference presentation hoping to take place this coming September. We have no direct competitors, and Stenoa's direct cost avoidance is greater than 50%, which would have saved the MUHC $300,000 in 2018 alone. Stenoa was born to change practice, offering a new standard of care, and its secondary cost effectiveness is exponential. Allow me to just give you a few examples of how we can take the data we've organized and leverage it further. We can use it for anatomical motion guidance in robotic procedures, predict premature stenoses, propose the most effective treatment for a unique stenosis, predict the likelihood of restenosis, and propose calculated angiographic follow-up periods. As you can imagine, the implications of this are revolutionary. Cardiovascular disease cost $555 billion in 2014 alone, and by 2035, the cost will rise to $1.1 trillion. Now is the time to act. Thank you very much, ladies and gentlemen. And on this note, I wish to invite you, together with Stenoa, to disrupt an industry.
Thank you very much, Jeremy. Nice presentation. Nice results. A lot of data. Diane, you want to invite the judges for questions, please?
Absolutely. So let me see if there are any hands up. Oh yeah, I see. Okay, Michael Mee, let's hear from you, Michael.
Can you hear me now?
I hear you, yes.
Great talk, Jeremy.
Thank you very much.
So you outlined a number of application areas for the technology, and that's very exciting. When you contemplated the regulatory path for this digital sort of AI-empowered device, do you foresee needing regulatory approval for each of those application areas?
Well, thank you very much for your question and allowing me to actually elaborate on that. Perhaps what I'll do is I'll share my screen again so that you have access to that slide, and we can go through some of them and discuss the specifics. But essentially, when we're protecting our intellectual property to allow Stenoa's algorithms to basically predict virtual FFRs, then that has numerous implications which won't necessarily need regulatory approval, but which will benefit from randomized controlled trial support, for example, or evidence-based support. And so by protecting the intellectual property with regards to and receiving regulatory approval with regards to Stenoa's virtual FFR prediction algorithms, well, these are the possible implications of how that data can be interpreted and can then be extrapolated and used in other contexts with the support of evidence from collaborators.
Okay, thank you. I asked the question because I'm trying to get a sense of the cost of driving to milestones to sort of empower clinicians to adopt the technology in these types of use cases.
Absolutely. Well, once we have this technology available and once it's actually on the market, then we're going to be working very closely with clinical researchers in order to work on these implementations and in order to provide the evidence for clinicians and interventional cardiologists to be able to implement them in practice.
Great. Thank you so much. I don't see any other questions unless I'm missing anything. I guess your presentation was very thorough.
I think Steve Arliss is his hand up.
My hand up.
Steve Arliss? Okay, great. Thank you.
Okay, look, I've spent 5 years as a director of a leading fractional flow reserve company called OpSense up in Quebec City. I find what you're doing is fascinating and potentially transformational. I understand that there are some non-invasive imaging solutions for fractional flow reserve that are in development, if not in commercial mode already.
Sure.
Please let me understand how your machine learning approach will be potentially more accurate and user-friendly than these non-invasive MRI approaches that I understand are coming.
Thank you very much, Mr. Arliss, for that question. It actually segues me to introducing you to a slide where we discuss some of the market competition, and I'd like to briefly address that with you in light of your question. So, as I mentioned in our presentation, we have no direct competitors that are offering exactly what we're providing, which is real-time guidance live in the cardiac catheterization laboratory for operators. However, what currently exists on the market is something called quantitative coronary angiography, which relies on simple diameter reduction and requires substantial observer intervention, but it's actually obsolete and it's even less used than FFR. Another competitor, which is what you're referring to, is HeartFlow. It's a very successful company based out of California that uses CT-guided FFR, so computed tomography-guided FFR. But it only offers pre-operative guidance. So when the operators are inside the cath lab, they're not actually able to measure the effectiveness of their restenting or of their reballooning and their revascularization process. As well, the patient's CT scan needs to be sent externally to California for processing, and our report needs to be sent back sometime later. Stenoa, on the other hand, is a local software, and it doesn't require any additional hardware. It operates in real time, and that dynamically segments coronary arteries, creating an optically consistent and clinically relevant picture for operators.
Wow, that's a terrific answer. Thank you.
Thank you very much, Mr. Arliss.
Thank you. I believe we had a question from Dan Roden next.
So yeah, my question was sort of again around the technology and the application. So as I understand, the pictures that you showed were really very, very cool in terms of outlining the coronary tree, but I didn't see data that sort of showed how you would predict this virtual fractional flow reserve. And so my question was, how big is the database with respect to how many FFRs you have? And have you validated it beyond the right coronary artery, which feels like the easy bed? And the hard bed is the left coronary system, which is harder to sort of dissect out and harder to analyze, I would assume. So sort of trivial questions, or not trivial questions, but sort of operational questions.
Sure. Thank you very much for that question, Dr. Roden. You're right in saying that we validated this in the right coronary tree. And the reason for that was effectively because we wanted to make sure that our algorithms that are dynamically consistent, again, that's a novelty of our product, are actually making their optically consistent segmentations properly. And we're currently in the works of validating this on the left coronary tree, which has its own challenges and encumbrances because of the overlap from the arteries, right? And I think that's what you're referring to. Sometimes you can have, for example, the LAD and some diagonal branches or marginal branches that will overlap with the circumflex, et cetera. And so what our algorithm is doing, which we're really excited about, is it's taking this 2D angiographic data and it's interpolating it through time, right? And the beauty of that is that with time, you're actually able to separate different features and you're able to isolate different branches and create different streams and repath out that coronary tree. And so that's what our team is working on currently, is validating this in the left coronary tree. And what we're going to be excited to present those results soon. As I mentioned, we're hopefully presenting at a conference in September.
Thank you very much, Jeremy.
Thank you, Dr. LaChapelle.
Thank you.
Great. So unfortunately, we're out of time, so we're going to move on to the next team, Ubenwa Health, and I'll ask Samantha Latremoy to take the stage, please.
So hello and good afternoon, everyone. I'm going to start today by just playing a few seconds of two nice, calm, relaxing audio recordings. So the first, and the second. Okay, so maybe that wasn't so relaxing, but I'm sure you all recognize that those are the sounds of babies crying, and I hope they've got your attention. They are indeed meant to disturb the peace to get everyone to listen. Crying is how babies communicate their needs. In fact, it can also tell us about their health. And you probably didn't realize that these are from two very different babies. The first is from a normal, happy, healthy newborn, while unfortunately, the second is from a newborn suffering from birth asphyxia. A newborn cry is actually an involuntary action directly coordinated by the central nervous system, and any injury to the brain can be reflected by changes in the cry pattern. So while you may not have been able to tell which of those cries were from a sick baby or a healthy baby, upon analysis of the waveforms, there are actually significant differences between the two. And we're using machine learning to harness those differences and use cries as vital signs to save lives and reduce disability rates. So what is this birth asphyxia? It's when a baby lacks oxygen at birth for a variety of reasons, and the consequences, can be severe, often causing various long-term disabilities, such as cerebral palsy, hearing loss, and learning disabilities. And in 10% of cases, it is fatal. Birth asphyxia is a serious problem. According to the World Health Organization, it's one of the top 3 causes of newborn mortality today, causing about 1 million deaths and over 1 million disabilities each and every year. And if we look at this world map of newborn death rates, we can see that there are far more newborn deaths in developing countries, like those in Africa and South Asia. The reason for the high mortality rate is that effective treatment for birth asphyxia has to be initiated early, so it requires a rapid diagnosis. In developed countries, early diagnosis of birth asphyxia is achieved by routine blood tests at birth and neurological examinations. But of course, this requires resources such as equipment and specialized personnel. But in low-resource settings like the developing countries, they don't have the right equipment, and births are not always attended by a doctor. And in these settings, asphyxia is often only suspected once visual symptoms start to emerge, such as lethargy or paleness. And at this point, it is often too late, and the baby has suffered significant brain damage. Clearly, there's a desperate need for a solution that is quick, easy, and inexpensive. And our user research really confirmed this need. Our founder, Charles, attended a pediatric conference in Nigeria just this past January, and he spoke with various health professionals, confirming what we had already expected. First, that asphyxia is the leading cause of newborn mortality in their respective centers. And in their experience, it's the lack of equipment and specialists that leads to the late diagnoses and ultimately causes the high mortality rates that they see. And finally, if a product like Ubenwa was approved as a medical device, they would be eager to put it into their practice. So our solution is the Ubenwa app, which analyzes an infant's cry sounds using machine learning to detect early signs of asphyxia in 3 simple steps. Having the solution in an app form makes it easy to distribute and to use. All it takes is to record the baby crying. The app will then detect if cry sounds were present, and then it will estimate the risk of asphyxia, all in about 10 seconds or less. So the Ubenwa app offers 4 key benefits. It detects swiftly, it is cost-effective, it is contact-free, the ultimate level of non-invasiveness, and it requires no clinical expertise, so it can be used by any and all birth attendants, no matter their education or their experience level. The Ubenwa app was developed based on our research. So if you remember back to our 2 samples earlier, while we may not have been able to distinguish which is healthy and which is sick just by listening, there are a few significant differences that more sophisticated analyses can detect. So using an existing database of normal and asphyxiated baby cries, we've discovered that asphyxiated babies have increased pitch, a greater instability in their fundamental frequency, and longer pauses. And we've used machine learning to take these key findings in order to develop our core technological components, which includes an algorithm to detect when an infant is crying, so distinguishing it from other noises, which currently is at a 96% accuracy, and an algorithm to detect asphyxia, which currently has an 88% accuracy. So we have very strong results so far. And we are currently acquiring new prospective data. We are working with partners in Canada and Nigeria at 6 different centers in order to recruit 2,500 patients over 18 months, which will be the largest database of clinically annotated newborn cries to date. We project that our clinical studies will end by June 2021, where we will be able to refine and validate our initial algorithms, but we'll also be able to explore new algorithms in relation to the data that we collect, which includes identifying the baby's needs and developing a disability prognostic tool. We will then be able to finalize our intellectual property and begin regulatory applications. We will begin regulatory clinical trials by December 2021, upon consultation, of course, with the appropriate regulatory bodies. And once we obtain regulatory approval by June 2023, we will begin the sales first in Nigeria, given our established connections there, and then we will expand to the full African market and South Asia, all areas with the highest newborn mortality rates. The medical device market in Africa is currently estimated at 5 billion, and it's projected to increase to 7 billion in 2023. And Nigeria alone actually makes up 22% of that market. Meanwhile, there's also a very strong trend towards mobile health globally, increasing to $332 billion in 2025. So medical apps are increasingly being accepted and used around the world. Our market entry strategy prioritizes rapid access for populations with the greatest need. So our primary approach is a partnership for distribution with a non-governmental organization, such as foundations that have a child health focus. Meanwhile, we will also take a direct sales approach by working with different governmental bodies and hospitals, and from there we'll be able to reach out towards the smaller clinics and birthing centers in the community. However, we are still exploring the best approach to this particular market of developing countries. We are confident that we are the team with the right skills to bring this forward, with backgrounds in computer science, machine learning, clinical research, biological signal analysis, software engineering, and app development. But more than that, we also have expert advisors from the fields of computer science, machine learning, and biomedical engineering to help us through. Before wrapping up, I'd like to take just a quick moment to discuss our vision for the future. So imagine a world where a newborn's cry is harnessed as a vital sign, where a mere recording of the cry can determine if they have birth asphyxia, autism, or are at risk of SIDS or hearing loss. In our ongoing clinical studies, we're tracking hundreds of variables across thousands of patients. And our vision is to build cry-based monitoring tools that will be used in both developed and developing countries to enable rapid interventions to save and improve the lives of babies. We'd like to thank all of our partners for their ongoing support. And we thank you for listening to us today, and we'll take your questions.
Thank you very much. Thank you very much, Samantha. Nice presentation. Nice that it's outside of the North American continent as well.
Yes, very much.
Diane, you want to ask the judges if they have any questions?
Yes, absolutely. I see we have a few hands up already, so we'll start with Dr. Hakim, please.
Presentation. My question to you is, does the early identification of asphyxia lead to improved interventions that reduce the damage to the health and the brain of the child? You have not shown any data that says early detection will improve outcomes of these patients, of these babies.
Yes. So currently the gold standard for the treatment of birth asphyxia is the initiation of a whole body cooling, so therapeutic hypothermia. And that needs to be initiated, or it's been shown that it has to be initiated within the first 6 hours of life. And actually in those who they initiated within the first 3 hours, they had even better outcomes. So the earlier the better. And the reason for this is, although there's a hypoxic injury, there's also a reperfusion injury that is going on afterwards. It starts at about 6 to 12 hours and it could go on for days. So if we don't get the intervention early, this continues to damage the baby's brain.
Thank you. Are there any other questions? Yes, Michael Mee, please go ahead.
Yeah, great presentation. Maybe a clarification question, and then if I'm understanding this correctly, a follow-on question. When you speak of this as for use in mobile health, are you intending the use case to be in the hands of parents on smartphones or in the clinic? And maybe assuming it's going to be more broadly distributed into the field, how do you sort of envision running clinical trials to ensure that that field implementation would sort of drive the same appropriate sort of determination that you see in a more controlled manner in terms of background noise, phone-to-phone variability, etc.?
Yes, so we are prioritizing this first as a medical device to be in the hands of a birth attendant, which could be the doctor, but a lot of times is often a nurse or a midwife or anybody else who's just specially trained, especially in Nigerian birthing centers, which are far out into the communities outside of the city centers. I'm sorry, I seem to have forgotten the rest of your question.
Yeah, I could speak to the second part of the question. Can you hear me, everyone?
Yes, I can hear you.
Can you hear me?
Yes.
Yeah, so the second part was about how we're taking into account environmental factors like noise and so on. And
Yes.
that's something we started building into our system early on, because the very first step of a system like this to work accurately is that you have to be able to isolate cry sounds from every other sound that could possibly occur in the environment. And as Samantha showed, one of the plots she showed, our cry detection algorithm is about 96% accurate, and we really spend a lot of time training neural networks in our case as well to differentiate cry sounds from a collection of about 100 audio samples that we actually pulled from different sources on the internet. And then in terms of our clinical studies themselves, we are doing them with hospitals because, as Samantha said, our target users are birth attendants. So it's not for homes right now. Some of the future products we plan could be used by parents, but this one is designed for that 6-hour period right after birth. And so all our clinical studies at the 6 sites are currently in hospitals, and that way we can make sure that we have clinical diagnosis of asphyxia for the patients that are considered to be asphyxia patients, and we have clinical confirmation of healthiness for the normal ones as well.
Hey, thank you very much, Charles. I'd like to call on Brent Norton next, please.
Sure. Thanks, and great presentation, Samantha. Have you thought through, you identified your customers and clearly who your patients would be, but you thought through how you're going to, the revenue model and how that's going to work and how you're going to control it in terms of when you go out to these customers and whether, the software could be reproduced in ways that you're not able to collect from?
Yeah.
Oh, go ahead, Charles.
Yeah. So this is definitely an area that we are still thinking about a lot. The best model to really have a good conversion from deploying our tool. One of the things we want to do is to work strongly with the public health system and foundations that are involved in a huge part of the delivery of care for especially mothers and babies in the countries. And so we are looking at a license for distribution model as one key model for revenues. So there'll be an entity that will be able to, because they have access to health centers, clinics that they run already. They would obtain a bulk license to be able to use that. And then because of, and then there's also a huge, for instance, in Nigeria, there's a huge private healthcare system. So many small private hospitals too, that we think would benefit a lot from just getting this package. Because the vision we see of the use of Ubenwa in the field is a setup where eventually the doctor can look at a screen and there are crib mics attached to the cribs of the babies. And then they can look at the screen and really just see the acoustic characteristics of each of the patients and get flags and get notifications of which ones are at risk and be able to act before that becomes irreversible.
Thank you. Thank you, Charles. I'd like to ask Stephen Arliss to speak next.
Thank you. And Samantha, I want to congratulate you. I think in my many years of competitions, I've never seen such a compelling presentation as you gave today. I really want to congratulate you. It was very compelling. My question is sort of related to, I think, Dan's that was given earlier. I mean, we all know the North American market and how revenue models are generated here. Could you just elaborate? I mean, technology is only as good as the potential solution and revenue that it's going to create around the world. So it's going to attract investors. So could you just elaborate a little bit more on how this is going to interest investors to support this and back it as you build it up? I think it's an incredible solution, but you need investment money.
Yes. So as Charles was mentioning, a partnership with a very good foundation with Child Health Focus is definitely our first go-to.
Okay.
But acknowledging sort of, it is a bit of a sticky situation, trying to generate revenue in developing countries in the medical device market. Because if they had all of the resources available to them, they would have the blood gas machines and they would have everything that they would need, and then there wouldn't be the cause for this sort of solution. We do really feel that the birth asphyxia is, in fact, just a really big stepping stone for us to be able to acquire data, have this large clinically annotated database of newborn cries that allow us to come up with other tools that can be used in North America and other developed countries, tools for parents, tools for the clinical sort of situation. So I guess we are also hoping that whoever might invest in us will also see this potential future. While there might not be a great amount of money in birth asphyxia because of the social impact sort of situation that it has, we believe that we can create a lot more tools from that.
Great answer.
Yeah, and to give two concrete examples of two possible tools that could result from our current work as well. So one is that one of the things that we are doing as part of the ongoing clinical trials is that we're asking the physicians to annotate data according to the reason for the baby crying. So one of the things we've seen already from research is that if the baby is crying due to pain or anger or sleep, whatever the cause is, there are actually alterations in those cry patterns. So we think that if we could ultimately collect this data and build an algorithm for that, this could lead to a new line of products for parents, as Michael had earlier indicated, that would really help new parents to give their wards better and more improved care. And then the second one is looking at a long-term prognostic tool as well. So we also know that today, brain scans are what is used to determine to have a view as to if this child will develop disabilities down the line. And because we know there's this strong connection already between the brain and the cry signal at that early age, because it's an involuntary response to stimulus, we're also looking at, we're doing a 2-year follow-up of all the patients in our study to say how are they doing. We're doing the Bailey score, which is a standard test for neurodevelopmental status of the baby at 18 to 2 years old. And we're doing this for all the patients in our study as well, so that not just are we building the birth asphyxia product, but we could end up with more insights, both for fundamental research, but for applied work that could be deployed eventually.
Thank you.
Thank you. Thank you very much. All right, great talk. I'd like to have MedSafer come in as the last presentation for today.
Hi, everyone. Thanks so much for tuning in today. I want to start by introducing you to Jeanette Smythe. Jeanette is my 97-year-old grandmother, and at the time this picture was taken, she was suffering from the side effects of taking multiple medications. She was feeling extremely weak and was complaining of memory problems, and she had severe anemia. Today, I'm going to present to you a software technology that our team designed to help people just like my grandmother. It's called MedSafer, and it's a solution that addresses the problem of polypharmacy, otherwise known as taking multiple medications. You see, polypharmacy is a massive problem. In North America, close to half of all older adults take 5 or more medications, and we've observed a 200% increase in polypharmacy in the last 20 years, which has led to millions of older adults seeking medical attention because of an adverse drug event. Polypharmacy and adverse drug events are having a major negative impact on the healthcare system. This problem has led to hundreds of thousands of preventable hospitalizations each year and billions of wasted healthcare dollars. And finally, this sobering statistic. Polypharmacy will be responsible for 150,000 premature deaths in the next 10 years. Now, if you're not already convinced that this is a massive problem for society, you should also know that with each medication that gets added to a prescription regimen, the risk of an adverse drug event goes up by 10%. And these are extremely serious complications that we're talking about. Taking more than 5 medications increases the risk of delirium. Taking more than 10 medications increases the risk of a fall by 50%. And this actually means that polypharmacy substantially increases an older person's risk of dying. But the good news is doctors have a solution to this problem. It's called deprescribing. Deprescribing is when an expert clinician carefully reviews a person's medications and stops some pills that over time are no longer needed or that may have even become harmful. It's a logical approach to the problem, but as I'm about to show you, it's far from simple. There are actually several barriers to deprescribing. First, while the evidence base is there and we know what meds need deprescribing, there are literally hundreds of rules from different professional societies. Sorting through these and knowing which ones actually matter and should be prioritized requires special medical expertise. And the fact is, this human resource is not universally available, not even close. Each person who could benefit from deprescribing has on average 10 medical conditions and takes 10 medications. Cross-referencing each of these takes time. And the reality is, many of these medications have accumulated over years, and some doctors may hesitate to stop medications when they weren't the ones to prescribe them in the first place. Makes sense. So, enter our solution, MedSafer. MedSafer offers something called software-facilitated deprescribing. This is a new healthcare process that responds to an unmet clinical need. And this approach to deprescribing will have a truly meaningful impact on the care of older adults. Our software is time-saving and simplifies the complex process of deprescribing. I'm going to show you that we have a demonstrated track record for success and clinical trial evidence of effect. And finally, we're already copyrighted. Let me show you how it works. When a patient is admitted to a hospital or a nursing home or sees their doctor in the clinic, their health information is recorded in an electronic medical record. Until now, this information has sort of just sat there, but we designed something called an application programming interface. So we can take existing coded healthcare data, process it, and send it back immediately to the doctor as a roadmap for deprescribing. And this is literally done with the click of a button inside of the electronic medical record. Now, if you're like me, you may be saying to yourself, well, that's nice, but what's the evidence that it works? Well, we have actually already used MedSafer to analyze the medications of over 6,000 people across Canada. In the results of this one study published in the Journal of the American Geriatric Society, we demonstrated not only that the software is safe, but that we were able to substantially increase the number of people with medications that were deprescribed with MedSafer guidance as compared to usual care. And the results were not subtle. We were 15 times more likely to stop sleeping pills, 25 times more likely to stop proton pump inhibitors, and this striking result. Receiving a MedSafer report meant that your doctor was more than 200 times more likely to stop dangerous antipsychotics that were being prescribed off-label to older adults for things like sleep. Now that I've hopefully convinced you that the technology works, the next logical question is, what's the market for a solution like this? Well, the market is vast. There are at least 4 sectors where MedSafer can be integrated into the prescribing process: in the community, in long-term care, in acute care, and in special populations. For example, we could integrate with a healthcare system such as Kaiser Permanente in the US. We've already integrated with a large EMR called Medicare, which has 5,000 beds, and with all of the long-term care homes in New Brunswick with over 4,000 beds. With a recent grant, we'll be integrating with a large electronic medical record called Point-Click Care, which has over 700,000 beds in North America. In acute care, I've already shown you that we've tested MedSafer on over 6,000 hospitalizations, but we also realized important cost savings. Now, some of you may be asking yourself, how does this not already exist? But the fact is, where deprescribing is happening, it's right now fully dependent on human resources. We're relying on pharmacists, expert clinicians, and things like static guidelines. There are only 2 other serious competitors, and they are still way behind us. Taper MD and Senator are applications that still require the manual input of data, and they haven't proven their effectiveness the way we have in clinical trials. Before I end, I want to take you through our timeline. And the take-home message here is that we're done our development. We're done our beta testing. We've published a trial. We're copyrighted, and we're already incorporated. In the short and intermediate term, we want to refine our revenue model. And while MedSafer is being used in 15 homes in New Brunswick and Ontario, we want to scale up across Canada and expand to other markets. But I'm not a business person. My colleague and I designed MedSafer. We're researchers. We tested it. It works. But there is a huge potential here that has yet to be realized. This software will generate a large amount of important data that will allow us to truly evaluate the value and safety of thousands of medications for a large portion of the population, especially if we incorporate artificial intelligence into the analyses. But we need a business partner to help us take this technology to the next level and realize our full potential. So to conclude, I want to take you back to where we started. Here's my grandma.
1 minute.
Okay. Here's my grandma again. Now that she was not part of the clinical trials that I told you about, but because I had access to MedSafer, I ran her information through the software and put in place a deprescribing plan for her. And yes, this story has a happy ending because just 4 months later, this is my grandma feeling much better. Her appetite was back, her anemia was gone, and she was feeling well enough to come out for brunch and enjoy some bacon and eggs. Thank you.
That's very nice, Emily.
Yeah, that's lovely. Great photo.
Yeah.
Great. So thank you very much. So we can begin the deliberation period. Are there any questions? Let's see.
You have analyzed, and because as you may know, many patients go to the hospital with one set of medication and leave with another set of medications that are completely different in many cases.
Sorry, you were cut off in the beginning, Dr. Hakim. Could you say the start of the question?
Yeah. Do you have any data? Have you been able to integrate your system with hospital records? Because in many cases, patients going into the hospital have one set of medications and leaving the hospital, they come out with another set of medications. I can tell you as a nephrologist, the average number of medications patients take somewhere in the range of 10 to 12. So I'm very interested in your process, but I want to make sure it also applies to hospital records, if at all possible.
Yeah, so thank you for the question. The main trial that we did was with hospitalized patients. So we analyzed their data when they are admitted to the hospital. And you're right, there are a lot of changes that occur along the way throughout the hospitalization process. We ran MedSafer on their home medications, and we were able to stop many of the harmful drugs that they had been taking at home as part of their usual care. I believe that if you ran MedSafer a second time before hospital discharge, you would also have an impact on potentially inappropriate medications that got added during the hospitalization.
And your system would allow that?
Yes, yeah.
Okay.
Great, thank you very much. So we have a question from Nicholas Franco, please.
Hi, Emily.
Hi.
Very nice presentation. Thank you. It's quite obvious what the savings on both sides for the patient and for the system could be. But what has been the feedback from the New Brunswick home or place hospital where you have implemented the system?
Yeah, so we've had feedback from a number of clinicians who have used MedSafer. In general, they find it very easy to use. It's a pretty integrated report. We didn't get to go into all of the details, but we actually have links to different materials that can help the clinician stop certain medications. So there's patient empowerment brochures, there's links to the most available evidence. There's studies that are incorporated into the recommendations. So for some medications where the evidence base is questioned, we provide some of the evidence to the clinician so that they can go and review it themselves. So in general, they've found it easy to use, up to date, and helpful.
And maybe one other question from a business model perspective. Is the app self-contained, or does it link to an algorithm database behind it?
So we have our own database behind it that we own, and the application programming interface can link with different medical records. I'm not sure if that's the question.
Yeah, that's fine. And in your discussions with some of the customers, what kind of business models have you been discussing? Because you can easily think of a fee-for-service type of model where you can actually take a percentage of their savings.
Yeah, I think the revenue model that's most developed for us is in the long-term care sector. So within different electronic medical records like point-click care, you can offer various supportive technologies that help with prescribing. And usually the long-term care home will pay per bed. So it's a per bed revenue model that we have. But you could also see if the government invested in this type of product that they may obligate nursing homes to make this available to their residents.
Thank you.
Great, thank you. So I'll ask Brent Norton to ask the next question, please.
Yeah, I was going to ask a similar question about point-click care. So it's per bed. I think you had a number of 700,000 beds. Can you give us a bit of the magnitude of how large that contract looks like, and whether it's renewable annually, or what the frequency might be, and how that relationship has evolved?
Yeah, so right now, when we develop in point-click care, we pay a certain amount for access to data, but it's a hunting license. So we would have to go after different homes in order for them to pay the added feature to use MedSafer.
Interesting. Okay. Thank you.
Great. Thank you very much. Are there any further questions? Nope. All right. Well, I guess that concludes the pitch section of our competition then. So what I'm going to do is I'm going to ask the judges, I'm going to send them an invitation to go to a breakout room. But I would invite the rest of you to please stay online. Dr. Freed will be speaking, and we have a special guest as well who will be speaking afterwards. So just bear with us for a minute or two, and we'll continue.
Thank you. And thank you, Kevin. And thank you for chairing this excellent session. Like everybody, I'm disappointed that we can't have a chance to see everybody in person, but I think the presentations were outstanding, and we thank the judges for their insightful comments and questions. When I reflect back in about 2013, when I was chair of the Department of Surgery, we had a vision to establish a surgical innovation program. The idea behind this was to build capacity in clinical innovation within our department. And in order to do this, the first step was to develop a graduate program in surgical innovation, a master's level program that would bring together clinicians, experts in business, and engineers from a variety of areas, including mechanical, electrical, software, and material science, and industry, and to bring them into teams in order to first identify unmet clinical needs. And the interesting thing about this was that the unmet needs were identified from the perspective of both experienced people in the fields, but also the refreshing view of naive people who would come into the hospital setting and ask the question, why are you doing this way? There's a better way. In that role, they were able to develop entrepreneurial skills. They would learn about intellectual property, regulatory and compliance issues. They would have an opportunity for real-life exposure by doing internships. And at the end, our goal was to have them develop solutions that ultimately could be commercialized. The success of this program was really rewarding. And my next step was to meet with Dean David Eidelman, and who's here, and I thank him for his support, with the goal of expanding this from surgery to all clinical programs, to make it a true clinical innovation program. And he was immediately supportive. We were able to expand our graduate program by taking in people from other clinical disciplines. And through David's support, we were able to develop partnerships with the community, such as with Dr. HAKIM, partnerships with industry, with government, and with a group of accelerators. The next step in our mind was to develop homes for the clinical innovation program. So immediately we saw the opportunity to use the Steinberg Center for Simulation and Interactive Learning, already a very innovative facility, to bring in companies that wanted to test their premises, proof of principle, et cetera, in an environment where there would be a large group of healthcare professionals at all levels to come through and give feedback. That was successful. And then our next idea was to bring clinical innovation into the hospital. We wanted to have a physical home for the program in the hospital for several reasons. One is it provided easy access to clinical key opinion leaders. We would be able to house startup companies there. It would facilitate clinical trials, and they would have access to labs for their research. In order to accomplish this, we applied for and obtained a strategic infrastructure program grant, and were able to build a center. And in fact, the official opening of that center was supposed to be today, but because of the COVID crisis, it will be postponed. In addition, working with Dean Eidelman and with his team, particularly want to thank Dimitra. We established this annual clinical innovation competition, really an idea that was brought forth by Dr. HAKIM. And with further partnership, MARIKA Roy through the MGH Foundation and the MI4 program, we've been able to expand the prizes. And I think that this is just going to grow forward. What are the next steps? We want to further develop these two spaces and attract tenants. We want to continue to build partnerships. We're about to spin off a digital health innovation program. We already have established a course on artificial intelligence in medicine, which is a great success. And our next real thing is to have a real commercial success that we could point our fingers to and attract investment. I have to say that the real success of this program has been that it's been due to the work and the leadership of Drs. Kevin LaChapelle, Jake Barlett, and Ed Harvey, and I want to thank them very much for that. We had invited Professor Remy Quirion to speak to the group at this point in time. Remy Quirion is a McGill person. He's currently, though, the chief scientist of Quebec in the Fonds de Recherche de Québec, Santé. And unfortunately, due to a last-minute meeting with the Santé Publique Québec, he was unable to attend, and he sends his regret. So he's asked us to relay this message to all participants. Innovation in the health sector is critically important for me and for the FRQ. The COVID pandemic demonstrated very clearly that we must innovate and improve our healthcare system. The key is research, science, and innovation, which is why the FRQ created a program of innovators in residence. A special call by the FRQ MEI MSSS generated over 700 proposals on innovations aimed at reducing the impacts of the virus. It is wonderful to see so many creative ideas and proposals. And I just wanted to add to that having our program in Montreal and in Quebec is ideal. We are truly a hotbed of information technology, creativity, outstanding academic institutions, and a robust startup community. And I think that with all these ingredients, we are bound for success. Our next speaker will be an example of such a success, Dr. Henrietta or Mimi Galeana. She's the co-founder and CSO of Saccade Analytics and Professor Emerita in the Department of Bioengineering at McGill University. The HAKIM Family Prize for Clinical Innovation in Healthcare was awarded to Saccade Analytics in 2018, its first year in existence, for the novel application of existing virtual reality goggles that can record eye and head movements. Based on their analysis, it became possible to generate health metrics that provide a quicker and more accurate evaluation of concussions and other neurological disorders, and to support rehabilitation methods. The idea originated from 30 years of research by Mimi in biomedical engineering, who retired as Professor Emerita in fall of 2019. Through her research, she understood the tremendous potential of eye and head movement analysis. So Saccade Analytics represents Mimi's lifetime aspiration to improve accuracy, clinical throughput, and patient comfort when diagnosing and treating neurological disorders, all at a lower user cost in even mobile sites. Her background is multidisciplinary, with undergraduate training in electrical engineering, graduate training in physiology and biomedical signal processing. The graduate studies followed a few years raising a family, and in the 1980s, she started her teaching and research career, focusing on quantitative models consistent with current neurophysiology. I'm really pleased that Mimi will join us today and tell us of the success of Saccade Analytics.
I appreciate the nice background you provided. And am I being heard?
Yes, you are.
Okay. So I was asked to give this presentation today to kind of keep the audience busy while the judges were going through the decisions they have to make. And it's going to be a hard one, I'm sure. What I thought I would do is, despite saying that our company has been a success, there have been some troubling moments, including a pandemic that made a mess of everything. So I just want to go through some of the issues of stress and getting along with your team and covering your money income. So I'll explain now in some simple terms. The first thing we wanted to do, given the research I had done, was to replace heavy, clunky, super expensive equipment in clinics that study vestibular and visual problems in eyes and head movements. They cost in the hundreds of thousands of dollars, and they're still sitting in our hospitals now. The other problem was throughput. It takes weeks to get an appointment, if not months, in some of these clinics. And the data, after you go to the visit, the data results come back sometimes a week later. So this is not a fast decision system to allow you to know if you are well or unwell. The treatment is delayed, and you're usually sent to many clinics. So our first goal was to create a package that would allow testing any eye and head reflexes and give the results on site right after the test. We achieved that, and that was the first big success, using VR goggles that are also instrumented with eye and head tracking in 3 dimensions. The second step was the diagnosis, or it's not a diagnosis, it's a metric we give the doctor who does the diagnosis. The second step is what you do with that data. And we chose the cloud, a SaaS, a software as a system approach, to analyze the data and provide the results within a minute. So now we had 5 to 8 tests that could be done on any subject in 5 minutes, all of them in 5 minutes, and the results of those recordings, coming back to the supervising person who's watching the subject, coming back within 1 minute. So this is 6 minutes total time. In and out. Patient knows what's happening when he's seen, and you can see a whole lot more patients than before. So 2 points to mark that are very important from our point of view. Mobility, mobile health. That's the first thing. So we can do this on the sports field or in a hospital clinic, whatever. But the other part is that the income for the company depends on the cloud service. So you have to have patients that are being recorded and sending data to the network, that otherwise you have no input, no money flow. I hope I'm making that clear. So our first big mistake that started to raise a red flag was at the beginning of the, what do you call it? When the pandemic was declared in early March. That's when the tap shut off. And we were faced with a possibility of maybe even having to go bankrupt because there was no money coming in. So choosing your revenue pattern has to take into account as much as you can. We never even thought there would be a pandemic, but it must, from now on, we all have to think as much as we can about what potential natural disasters or this accords between countries or something are going on, and plan so that you are robust in the presence of these disturbances. So we missed out on the business that you need patience to make money in this case. So the other issue is okay. So until the pandemic started, we did achieve the tools we wanted, the analysis we wanted, the installations on the web, everything would have been fine. We already had a network of clinics in 2018 and in 2019 across Canada, from BC through Quebec, and there's one in the Maritimes. We had a large number of team sports like the NHL and other clinics in the hospitals that were using our equipment regularly. So things were working properly. And from that, we were gaining a big database on the cloud from which we could perfect the coding and the detection of anomalies confirmed by the doctor. So things were rolling along very well. And then the pandemic hit. It hit while our CEO was in Toronto with planned meetings with venture capitalists because we were doing so well, and new clinics throughout Australia. Again, the tap shut because the meetings were all canceled. Nobody showed up except our CEO and her friends. So we've been trying to find a new path to last over the past couple months. While we're in quarantine, we continue working. And with the help of the government that created these furlough refunds so companies could keep their people at a lower cost, that allowed us to develop a new direction, a direction that did not require live patients to produce data. Instead, we created a mobile app. Again, mobility is always an important issue that would help binocular and head coordination in a rehabilitation environment using your mobile phone. So that is doing very well too. Australia is going to distribute it to all its rugby teams. So we're back on, even if there's no patients allowed to use our equipment. the lesson to learn here is don't have only one revenue flow. Even though it's doing good in the world, it won't work if the flow is interrupted for whatever reason. So now we have two flows. One is healthy, sick people with a mobile app at home, play with it whenever they like. And when they see the doctor later, they'll judge on the improvements or not. And the other is the raw data from clinics or team sports after injury. So they back each other up. So the main thing I want to emphasize from this is that you have to be ready without going crazy. We were on the edge to face surprises, things you would never expect to happen and that might happen that might affect what your company can do in the future. So you have to be very flexible. You have to let your team give ideas, especially when you have a small team and they're young and they're energetic and they don't want the thing to fail. They will come up with all kinds of things. And sometimes you have a huge surprise, like the mobile app. I never would have thought of that originally. So all I can say at this point is that after hearing all those teams for the competition today, I am really in awe at what the kids, because for my age, they're the kids, are achieving in all diverse areas. The committee must be having a very hard debate at this point. I'm sorry I can't answer questions if somebody had any, but I can tell you that Saccade Analytics is healthy.
Actually, if you like Mimi, we could do a little bit of questions. If anyone has any questions, I'd be happy to do that since we have a little bit of extra time. So
okay,
yeah. So if anyone has any questions, please just raise your hand and we'll see. We'll call on you.
So I have a question.
Great.
Hello, David.
So hi, Mimi. So you made a huge transition from an academic world to the business world.
Mm-hmm.
I mean, I just listened to you talking about having revenue streams and so on. This is quite a different way of thinking. And you gave us a lot of cautionary tales. So could you tell us a little bit about that transition? Because although some of the younger people are at the beginning of their careers, they haven't declared themselves as either academics or not. Several of the people who presented, like Stella and Emily, are academics. And so I would like to hear a little bit about your experience.
Okay. Let's see. At the beginning, I wasn't the chief science officer. I was more like a director on the board and an advisor when they had questions and how does this reflex work and is there any literature on it? So I was still acting like a supervisor for a graduate student, only they were not graduate students, but they were very smart and they wanted to learn. When it came time to go to the FDA, to go to Canada, Health Canada, etc., and figure out a revenue, whether or not you could make money doing this, what pricing should you use? I'm sorry, but that is not what I like. No, what I like is to see a benefit to the population, to people. So I gave it to my daughter, who's nearly got a PhD in economics and loves this sort of thing. She's the one that was off in Toronto and who got the contracts and that in Australia for all the rugby teams in the doggone continent of Australia. I mean, I'm very impressed by her, but I solved the problem by passing that to somebody else.
So the message is, if you're an academic, don't even bother.
Well, some academics love running a business, but I'm not one of them. That's all. But you're right, though. It's
and here I thought all my PIs were really just entrepreneurs whose clients were, whose market was CIHR. And,
yeah.
And
well, it's a different viewpoint you're exploring then. You don't have to document that it'll work.
Well, that's true. It's almost a negative. If it really works, then they don't want to know about it.
Yeah, they'll need the money. Okay, does that answer your question? Okay.
It does.
Great, thank you so much. So Dr. Freed has a question. So Dr. Freed, please go ahead.
So first of all, I would like to thank you for your presentation, and it was totally honest, and I think was very important for people to hear that. Maybe to take the devil's advocate to my dean, at my great risk, of course. I think the reason I believe that Mimi went into this, there actually wasn't because she wanted to necessarily make a whole lot of money, but to get her idea out there to benefit a lot of people. And that's what innovation allows us to do. But in order to do that, in order to commercialize it, you need to have a sustainable business model in order to get it out there. And what we're trying to accomplish through our innovation graduate program is to teach clinicians that it's not a dirty word to take a really good idea. And they have good ideas. That's where they learn that in their clinical training and in their research. But to take an idea and be able to disseminate that widely through innovation. But in order to do that, it takes certain skills that we don't all have. So either you could just take your idea very early on and pass it on or commercialize it or license it to people that are used to doing it, or to develop a team like the type of teams that I've presented today that have expertise in healthcare, that have expertise in business, have health expertise in engineering. And as a team that's greater than the sum of the parts, they can make a successful enterprise. And I think that's a very important lesson. So I applaud you for it, and I think I thank you for your time.
Lost you.
That's all. That's all. Yeah, that's it.
Thank you.
Thank you so much. Does anyone else have any questions? Okay, I don't see anything. So what we can do is just take a little 5-10 minute break to stretch our legs while the judges deliberate. And so I would suggest coming back for 7 o'clock, please.
Our deliberations, the judges and the numbers are in, and we would like to present the winners of the awards. And I'd like to introduce Steve Arliss, who will talk about the judging and our deliberations.
So thanks, Kevin. And look, this is my third CLIC HAKIM Prize jury process. And I must say, with all honesty, this was the most interesting. And we had a panel of 9 judges. And despite some differences in the way we saw individual pitches or presentations, I think we came out of the whole process feeling good about the outcome. But it's interesting, when we do announce the first prize winner, the HAKIM Prize winner, it's interesting. I don't think I've ever in the 3 years seen the majority of judges pick one presentation as number one, but that happened this year. So when the number one award winner is named, I want everybody to know that that award winner was well-deserved and clearly shone in the eyes of the judges overall versus everybody else. But there was some spirited debate about the prize for the Roy, and we ended up with an interesting compromise solution, which Kevin will talk about. And I always love being part of a jury where we come up with some creative solutions that keep everybody happy and blood off the floor. So on that note, Kevin, I'll pass it back to you.
Thank you very much, Steve. And I'd like to thank the judges for the time. And I have to say, the judges really put a lot of effort into the judging process and the preparation and the comments as well. So I think the participants can be rest assured that it was done at a very high level and everything was really taken into consideration. So I'd like to thank the judges for that, and again, thank the participants for the excellent presentations as well. So I need to present the MARIKA ZELENKA Roy Prize, and that's founded by Madame Roy, along through the foundation of the Montreal General Hospital. She is one of the first female graduates of McGill University in electrical engineering. And she has seen in a vision and the importance of innovation and teamwork, and has graciously funded this prize. And I'd like to announce the winners of this prize, because as Steve pointed out, we came up with some innovative solutions, and we felt that there was not only a numerical, but also a statistical tie between the following two competitors, and that was GyroClear and MedSafer. And therefore, we would like to award them both this prize. Now we present this issue to Dr. David Eidelman, because now it's going to be, I think, his problem to figure out how to do this.
Yeah, I had a feeling that was coming.
We do not feel that the prize as a monetary sum should be split.
I think I have to start solving problems like this too. I'm just going to decide that I've solved the problem and say that it's Dr. Kevin LaChapelle's opportunity to find a solution.
Right. It's like the Olympics. When it's a tie, you both get the same color medal. You don't get half a medal. So anyway, I'd like to congratulate those teams on that excellent, excellent finishing. And I'd like to give a round of applause, even if it is virtual, to both of them. So great work to both of you. I don't know if we can see them, or you can clap or make noise so we can see your face. GyroClear and...
Thanks very much.
Yay! Yay!
Thanks, everyone.
Thank you.
Is GyroClear there?
Congratulations.
Yes, we are here.
Thank you very much.
There you go. Okay.
Really appreciate it.
Congratulations, team.
Great work. Great work.
Great. Okay.
Last one, babe.
Yeah, I'm coming up. I just want to make sure I get the right one, you know. So I'm very happy to present this year's winner, the HAKIM Family Prize in Innovation. And as Steve said, there was a lot of agreement on who would be our pick. And so the winner is Stenoa.
Wow, I'm incredibly honored. I don't even
Congratulations, Jeremy.
I'm incredibly honored. I don't even know where to start.
Congratulations.
Hey.
Very good. They were all really close, really, really close. Great presentation.
Great job.
I'm incredibly honored. And if I could just give a few words to the judges and to everyone, all the other competitors, you guys were exceptional. You guys were so incredible. And I can't believe this. Everyone deserved it. And really, this was an incredible honor. I think you're going to hear my mom screaming in a second. I'm sorry about that. I'll just give a bit more information about myself. I'm only a first-year medical student at McGill, and working with these four people here, Dr. Ivanov, Dr. Spaziano, and Tomer, this was an incredible honor. This was an incredible privilege. And these people are superstars. They deserve to be recognized. And so I just want to put up a picture of us so you guys can see who they are. They are superstars. They have made this project happen from start to finish. And I'm truly speechless. It wouldn't have been possible without any of them. And really, to the judges, thank you so much. We're going to make you proud. And let me just pull up this picture so you can all see it. I'm shaking. Just give me a second. Here we are. And
if I can just say something.
Yes, please.
This is Steve Arliss. I've been a medical entrepreneur for my whole life, and I want to say you have a great potential with what you're doing. It's a game changer. But what I would give you as advice is surround yourself with a couple of people from the business world that understands how to commercialize this enormous potential of this technology.
Most definitely, Mr. Arliss, we will do that. And with the opportunity you've given us, we will make sure to make you all proud. Marco, Dr. Spaziano, Dr. Ivanov, and Tomer, you guys have been the best team. And I'm confident that together with the right business partners, we're going to make this a true success. So thank you so much to Dr. Fried, to Dr. LaChapelle, to Dr. Eidelman, and all the judges and all the organizers, and Mr. Hakim, Dr. Hakim. It's been an incredible privilege taking part in this competition, and you've given us a tremendous honor to receive this prize, and we'll bring your name good reputation, hopefully. I don't come from a medical family. I'm the first student in medicine in my family, and it's been an incredible honor working on this with everyone. All my mentors throughout this journey have been just incredible, and I'm speechless to all of them, from the beginning in basic science to clinical world now, and I look forward to seeing this project become a true success with all of your support. So thank you, Mr. Arliss, thank you to all the judges, and thank you everyone for your support of Stenoa.
Great work, Jeremy. Congratulations
thank
again.
You so much. Thank you, Dr.
And I don't know, Dr. Eidelman, would you like to say a few closing remarks, and perhaps Dr. Hakim as well?
Well, for my part, this is my third time being at one of these, and I have to say it's always amazing the extraordinary innovation that goes on, and I'm glad I'm not a judge, because it's really tough to pick winners. I want to congratulate all the winners. I want to thank everyone who worked on it, especially the communications team, obviously Kevin and Jake and Jerry, but also I want to thank all the people who took the trouble to put in their applications. Thank the judges for all the hard work they did to put it together. And I want to finally thank Dr. Hakim for having the vision to push us in this direction, and he's created something really amazing. So thank you very much to everybody. Have a good evening.
And thank you, David. Dr. Hakim, would you like to say a few closing words?
Again, I'm very proud of all the competitors and all the work that went into this competition. And I have to say that it took us a lot of back and forth in the judgment phase of deciding between second and third, and then we decided let's just give them to both second and third. But I want to assure the fourth and fifth that the scores were so close that you should not be discouraged by the fact that you did not win on this phase, because all 5 projects were amazingly strong and very potentially very helpful in improving healthcare for a lot of patients, and we work for them. So I hope those who did not win will continue to work on their project, because it's still well worth it. Thank you.
Thank you very much, everybody. And I would just like to underline on the last slide the supporters of the CLIC, and in addition to the cash donations and the prizes, you have the Simulation Center membership and support, FASCN Startup Program, which is part of the legal and component to your program, Novo, which also provides some strategy and product development, and you have access to other professional programs, and arrange a US tour with the Dobson Center, and of course free access to university facilities and services, which many of you already have anyway, but you're going to get it super free now. So again, I would like, on behalf of Jake Barolette and Diane Wiedner and Sarah Stack, I'd like to thank you for attending, and I hope to see you all next year. Thank you very much.